The orderly program of events in a cell's lifetime is known as the cell cycle. The eukaryotic cell cycle is divided into two phases: a brief mitotic phase, in which a cell divides to form two cells, and a longer period between divisions called interphase. Interphase can be divided into three distinct phases: G1, S, and G2. The G1, or first gap phase, is typically the longest phase in the cell cycle. Most cells will grow in size and manufacture a number of different proteins during G1. At the appropriate time, a molecular signal will cause the cell to enter the next phase of the cycle. During the S, or synthesis phase, the cell replicates the DNA in its nucleus in preparation for cell division. Precise control of the molecular machinery that replicates DNA ensures that only one copy of the cell's DNA is made during the S phase. After the cell has replicated its DNA, it enters the G2, or second gap phase. During G2, the cell may continue to grow and produces a number of molecules in preparation for the upcoming division. When the cell is ready, a molecular signal will cause the cell to enter the mitotic, or M phase of the cycle. During mitosis, the cell's DNA condenses into visible molecules called chromosomes. The chromosomes are carefully sorted between the two forming cells so that each cell receives one copy of every chromosome. At the end of mitosis, the cytoplasm is divided between the two daughter cells in a process called cytokinesis. Each of the daughter cells will enter the G1 phase of the cycle. Different types of cells exhibit variations of this basic cell cycle depending on their function in the organism. Some examples of different cell cycles can be found in the Variations section. The progress from one phase of the cell cycle to the next is controlled by a number of proteins in the cell. To learn how these proteins direct the cell cycle, DNA synthesis, and the events of mitosis, go to the Advanced section on the molecular control of the cell cycle. Uncontrolled cell division in the body can have harmful effects, leading to the growth of tumors and cancer. In the Cancer section, the role of tumor suppression genes is examined as they relate to the cell cycle.